Absolutely. Hopefully everybody can hear me. First of all, I apologize for not being able to be with you. I had to teach a 600-person class 5 hours ago, and I just couldn't find a way to quantum teleport all the way across the country to see you. But hopefully you guys are working on that. So today what I'm going to talk about is a project that I'm leading in collaboration with Grant Roscoff at Stanford University and also Norm Tubman at NASA Ames, who many of you may know, which is aimed at being able to create ways of modeling biochemical reactions on quantum computers for the first time. And so what we're doing, what we're thinking about is how do we come up with a combination of machine learning and quantum computation that allows us to model biochemical reactions with high accuracy, but also with relatively high efficiency. So the motivation for this work are different kinds of biochemical species, as I'll show here. So this is an example of a prenavir. And so for decades, people would study different kinds of therapeutics like a prenavir using things called QMMM. So for those of you who are not familiar with QMMM, QMMM stands for quantum mechanics molecular mechanics simulations. And so what these simulations would do is that they would treat a region around a particular chemical species like this one as a quantum mechanical region and embed it in a larger molecular mechanical, classical mechanical region. So for example, all of these purple residues, this happens to be HIV1 protease, would be modeled classically. And so people have used this for decades and decades. It won the Nobel Prize in 2013. But the problem with this technique over the years is that it's not necessarily accurate and it's certainly not very fast. So many companies over the years have used QMMM to design molecules like this. This is an example where they use QMMM to exactly match the HIV1 protease binding pocket so that they can create a drug that just fits right in there. But as I was saying, QMMM is very expensive, very expensive, and it creates a lot of problems in terms of generalizability and being able to actually model a wide variety of things. So even though QMMM is used for up to 20% of all drug discovery right now, it's way too expensive and is actually holding us back in a variety of ways. And so we asked the question, what can we do that's actually better and perhaps also incorporate quantum computing at the same time? And we use these kinds of metrics just to understand kind of where we're at with QMMM. So if we wanted to model a large protein, let's say for understanding proteases or kinases, you have to basically model thousands of atoms. So you'll have waters in a box with that protein. And just as a reference point, if you wanted to do the quantum mechanics on just one single point of that system, let's say 50 atoms, that's going to roughly take you 2 to 10 core hours using reasonable codes. If you want to scale up to a full QMMM simulation where you're going back and forth, you're doing the quantum calculation, iterating that with the molecular mechanics, going back and forth between them, that's basically going to take you 7 days up to weeks to be able to do. We've done a lot of these simulations in my group. They can take a huge amount of time. And so what we ask is, can we do better? And the answer is yes. And so what I'm showing you is a simulation right now of ATP hydrolysis using high accuracy quantum computing and electronic structure data intertwined into a machine learning framework. And so the basic idea is that we can try to do truly quantum MD by actually replacing the QMMM with quantum computed results that will feed into a molecular dynamics force field. We can literally train a molecular dynamics force field on quantum computed results to produce something that's both faster than QMMM, but also more accurate in the quantum mechanical regions. So that's what we've done. That's the goal. That's sort of the punchline. Let me tell you how this exactly works. So as I was saying, for many years, people have thought about using QMMM and used it quite successfully to model a variety of reactions. But the problem with QMMM is not only that it's slow, but the fact of the matter is that it's actually very hard to define these QM regions. So you can say that maybe they're over here or they're over there, but then you have to also match the boundary conditions around those regions. And if a reaction spreads throughout a larger system, then you have to think about how do I adapt my QM regions. And so this causes a lot of problems. There are also issues with modifying older force fields to work with the QM. And so this is historically challenging. But what we think to be the solution to this is to actually use machine learning to develop force fields. And I'll be clear, I've spent time working on this for years. This isn't a new thing. So basically, since about 2007, and perhaps there are instances before that, people have been thinking about how do we parameterize molecular dynamics force fields using machine learning. So can we just make a machine learning force field? And as it turns out, those kinds of force fields are highly efficient. So one of the first examples of this were the Baylor-Parinello neural networks, very old version of machine learning, where what people would do is they would take in different molecular coordinates, so the positions of, let's say, oxygens and hydrogens in water. They would apply symmetry functions and then have neural networks essentially learn the energies of these different kinds of atoms. And so this earlier network actually transformed much of, I definitely say engineering and increasingly biology by allowing you to construct a machine learning potential that's, again, going to take in molecular configurations, give you an energy. And because things can also, the neural networks can be trained on forces or differentiated to obtain forces, you'd also be able to get forces out of this. And so these neural networks have become incredibly powerful over recent years. They've grown in scale and complexity. And so now we have very, very accurate types of neural networks. One key example is MACE, and I'm not going to argue that this is the best network. It's just one of the networks that we've been using. And so this kind of neural network, which does essentially the same thing I described before, is much more transferable, much more generalizable. And so MACE is an equivariant message passing neural network, much more sophisticated, uses message passing between different nodes in order to communicate the information, and it's equivariant over translation, rotations, and other kinds of symmetry operations. And so these networks can now readily learn force fields for molecular systems. So it could be biomolecular, or as I said before, it could be different kinds of systems from chemical engineering. And these force fields, very importantly, once they're trained, they can be 1,000 times, more than 1,000 times faster than previous molecular mechanics models. So instead of having to consistently reevaluate forces and take derivatives and propagate Newton's equations, in some sense, you can train these networks to very quickly predict forces. And so you can get around some of the really costly aspects of using previous molecular dynamics. What's also important about these networks is that since you can train on energies from quantum mechanics, now you don't even need, let's say, a QM region if you're modeling something in QMMM. What you can literally do is train your force field to be incredibly accurate up to the level of accuracy of the quantum mechanical solution that you're obtaining. So if you wanted to train these networks on DFT, great. I'll show you some examples of that, easy to do. That's what most people do. But you can also train these up to really high accuracies. So if you wanted to go to couple cluster, I'll show you examples of that. But you can go to DMRG, you can go to any kind of quantum mechanical solver you want, and your network will have that accuracy essentially automatically. And so hopefully what you're getting from this is that given that you can have these kinds of networks, now you can do very interesting things and potentially include quantum computing. Now, the question you might ask is, you don't want to train these networks on quantum computed data. There's just not enough data. Quantum computing is expensive. Absolutely right. And so one way that we can get around this, and this also works for higher levels of electronic structure, is that we can train these neural networks on DFT, or perhaps even before that, we can start with foundational models. So let's say MACE0, and then we can retrain them using DFT data for your specific system. And then if you're smart, you can then transfer learn to a more accurate theory. And the reason why you'd want to do that is A, obviously you do want to train to a very accurate theory, but you want to do so at less cost than, let's say, retraining the entire network. So with transfer learning, you can train on highly abundant, but maybe low cost data like DFT data first, and then you can transfer learn relatively few samples with much more expensive data. So maybe 10 to hundreds of samples of either high accuracy electronic structure data or, as we'll do here, VQE data. And so this lets us create networks that are extremely accurate, but to do it relatively quickly. We don't need that many points from a quantum computer or higher level electronic structure, but yet we can obtain the same kind of accuracy for our quantum mechanical regions. And so what we realized is, we can put all this together, and what we're adding here is that we can use quantum results from quantum hardware to do this training and produce force fields that are fast like ML, but potentially accurate like quantum computers, or at least in principle, the quantum computers of the future. So our workflow is the following. So what we do, like I said, is first we train on classical electronic structure data, and then we fine tune through transfer learning on much more high quality data, like data from VQE. And I can go into why we chose VQE. I'll get to that in a second. Once we have that data, we can machine learn the kind of neural network we need to describe all of the interactions among all of the chemical species to create a potential energy surface with forces. And then once we have those surfaces and those force fields, we can run any kind of molecular dynamics that we want. So you can do plain vanilla molecular dynamics where we just solve Newton's laws. We can do accelerated molecular dynamics, maybe where we steer it, or we do umbrella sampling. We can do metadynamics. We can do whatever your flavor of accelerated sampling is once we have those different kinds of force fields. So that's kind of the setup that we've created. Let me show you just a few examples. So the first test case that we did, and I'll come back to this when we talk about our quantum computed results, is just thinking about water. And I'm showing this not because water is difficult or exceptional or something like this. It's just that you're probably familiar with the way that water should work. So what we can do is we can first train on hundreds to thousands of samples. This was sort of early days. We generated many samples and 16,000 different configurations of just water. So those configurations are going to have stretched bonds. They're going to have stretched angles. And so just be a variety of different configurations that we would obtain from, let's say, PBO0, density functional. And then once we train on that, we can transfer learn to VQE data. And so I should say for this initial work, for these results, I'll be showing you simulated results, quantum simulated results. I'll show you what we're getting on actual quantum hardware shortly. So what I show here are what are the potential energy curves we get from doing this. And so if you look on at least my left-hand side, what you see is the green curve. The green curve is our neural network trained on density functional data. And we know that that potential is not exact. We know that we can get more accurate things. And in particular, the yellow curve, this full CI curve, is essentially the exact curve. That's the curve that we would want if we really had high accuracy data. So we can first train the green curve, and then we can transfer learn to get the blue curve. So we can try to transfer learn using select high accuracy points. And when we do that, we do in fact recover the yellow curve. What you can see here is that I do show error bars, and I show that on purpose with the blue curve. So you might say, oh, it's not looking perfect. I'm showing that on purpose just so that you better understand how the method works. The blue shaded regions are regions of uncertainty. And so we actually use active learning within this framework to then further train our force field on the points where we see the greatest uncertainties. So if we were to continue iterating on this particular potential, we'd pick some points at the bottom, at the minimum here, and at large bond lengths to try to fill in and better learn if we wanted to learn the full surface. So we can do this for water, and we can fill in VQE results, which is what we did. And once you do that, you can actually obtain, nice little movies of the dynamics. So this is just two sets of water, and this was trained on VQE data. And so VQE simulated data. And so you can see that you can get pretty much exact dynamics from doing this. But what you might say is, well, that's nice, it's water, it's not biology. So the biological target that we're thinking about right now is ATP hydrolysis. So many of you might not be biochemists, you might not appreciate this, but ATP hydrolysis is the reaction that essentially powers our bodies. That's the reaction that releases about 7.5 kcal per mole per ATP breaking. So in hydrolysis, what happens is that you break the last phosphate group of ATP to produce ADP. And as it turns out, a lot of diseases are related to the activity of the enzymes that help with that breaking, that ATP hydrolysis. The hydrolysis itself has a very high barrier, so typically you need enzymes to make this happen. And we have something like 10 grams of such enzymes in our bodies at any given time that are just helping with this kind of hydrolysis. And so one type of enzyme that tends to have up or down regulation in cancer is the ATPase enzyme. As you might imagine, if you have cancer, you may be breaking ATP more to release more energy to do more reconstruction and grow more cells. And so we wanted to target and model this ATP hydrolysis reaction. Historically, it was actually, there are many questions about how this reaction worked. It was one of the first models to use QMMM that I mentioned at the beginning. So actually R.E.A. Warshell, who won the Nobel Prize for QMMM, he spent much of his life trying to understand ATP hydrolysis. And so we wanted to attack just that problem. Can we model that accurately using these kinds of force fields at much, much, much less expense and even more accuracy than, let's say, QMMM or REA were able to do beforehand, so that we can understand how this hydrolysis reaction works and potentially drug it. So as I was saying, this is the reaction that's sort of at play. This is a little bit clearer sketch. What we're trying to understand is this bond breaking right over here. So you see this R1 coordinate. What happens is this phosphate group over here is going to break off at that point and produce this ADP. So ADP is over here, and it's going to release this fragment. So we want to understand exactly what happens there. And what people previously hypothesized is that this reaction could occur using one or two main pathways. So what could happen is that this PO bond just breaks, so this R1 distance just grows. And if that R1 distance just grows, that's called the dissociative pathway. That would be the top. But people were also aware of that there could be something called an associative pathway where water comes in, and this water, just the presence of the oxygen, kind of causes this P to want to break its bond with the oxygen, the other oxygen. So this is called an associative pathway. And for decades, people actually weren't sure, and QMMM had a little bit of a hard time trying to resolve these things. People could never exactly get the 7.5 kcal per mole free energy difference that I just said is released during this reaction. They could never actually simulate that and get that right. And they had a hard time actually getting the barrier for this reaction, so how much energy it would take in order to break this bond, to initiate the breaking of the bond. So we wanted to answer that. And so to give you an example of just how this framework works, and it's all up and running, what we did is we modeled using this machine learning framework over 1,600 atoms. So this is the ATP molecule in waters, and there are a couple of ions. So there's a magnesium and there's a sodium ion. And we modeled this by training MACE. So we train MACE to two levels of theory, actually. So I showed you the MACE architecture briefly before, obviously a little bit complicated, but predicts the energies and the forces. And we trained this in two rounds. First, we trained it from its foundational model to what's called a meta-GGA functional, which is pretty accurate. And then we trained it even further to a hybrid functional, and most people would say hybrids are very accurate functionals. They're indeed very expensive functionals. And in doing so, we obtain a force field that's very, very accurate. So you can see the RMSEs for the energies and the forces here, and they're relatively small. So we're talking millielectron volts per atom or per angstrom. And so that's extremely high quality, high accuracy force fields. And so doing this, we can literally see exactly what happens. So once we have this force field, we can run all kinds of dynamics, as I said, and obtain free energy profiles. And we can resolve exactly what pathway this reaction went down. So if I look at these figures here, I can look at the barriers. So this is the free energy as a function of the reaction coordinate. And we can see that this curve here has the lowest free energy barrier. We call that the unbiased pathway. It turns out to be what we call a concerted pathway, halfway between the dissociative and associative pathway. So the first time using these kinds of force fields, we can actually see this reaction occur, and we can resolve the free energy surfaces to really high degrees of accuracy for the first time. And so we did this using DFT data, and I'll tell you right now, we're now using quantum computing data. Maybe a smaller example where I'll show you the quantum computing results today is metaphosphate hydrolysis. So one can imagine, can we do a smaller mimic? As you know, it would be hard to get that full ATP, 1,600 atoms on a quantum computer right now. I'll tell you how we're approaching it in a second. But we can obviously start with something that's smaller. So one similar reaction is called the metaphosphate reaction with water. It also undergoes hydrolysis, maybe a little bit differently. The basic idea is that this metaphosphate group is going to break a hydrogen-oxygen bond here. So this hydrogen will end up with this oxygen, and the OH will bond to the P, and that's also called hydrolysis. You're breaking the water. There's also a two-water mechanism where actually the hydrogen shuttle between two of the waters, but maybe we won't worry about that today. The reason why we looked at this reaction, as I'll show, is that after a variety of different tricks, we can actually get this reaction down to 44 qubits. And in particular, this one water reaction, we can get this down to about 44 spin orbitals, which is 44 qubits. Takes a little bit of tricks to do that, but we can model this accurately with just 44 qubits. And so once we do that, what we want to understand is what is the pathway, just like before, for the ATP hydrolysis. And we can train our neural network and then understand what pathway there is. So we did this, and this is using first DFT data, and then retraining up to couple cluster UCC data, and then finally quantum computed data. And you can see that we can model this hydrolysis reaction with two waters. So this is kind of a slow simulation, but at the end, you can basically see that one of the waters is going to break and form an OH bond to the gold phosphorus atom. So there it goes. And so we can actually see that reaction happening with quantum computed data. So I gave you an overview of how this happens, and obviously, there's sort of a handshake between the quantum and the classical HPC. The classical HPC is really helping with the machine learning, but let me tell you a little bit about how we're doing the quantum, and this is ongoing work. And I have to thank NERSC for giving us the time to actually do a lot of the quantum simulations. So how we're modeling the different quantum species at the base level is using the adapt algorithm. And specifically, we're using what's called coupled exchange operator adapt, which has a specific pool of operators. So if you're not familiar with the adapt algorithm, the basic idea is that you start with an operator pool, and you start with an initial wave function, typically a Hartree-Fock wave function. And then what you do is you pick specific operators that are going to decrease the energy that's associated with that wave function. So we're going to use the variational principle here and say that the best wave function, the best ansatz that we can use is going to be the ansatz that gives us the lower energy. So in adapt, instead of taking all the operators that you might get, let's say from a couple cluster ansatz, and that might be many operators, instead we selectively choose the operators such that they lower the energy the most. And so we'll choose operators, we'll add those to the wave function, we'll re-optimize the parameters, and then we'll evaluate and see if we're converged in terms of energy. And then if we're not, we'll add more operators and continue to try to converge the energy. So this is highly efficient, and I should say many people ask, do you get barren plateaus? No, we don't. We've never actually seen this with the adapt algorithm because of the fact that we're carefully selecting parameters and operators. And so to give you an example of this at work, if we just did straight up VQE based upon a couple cluster ansatz, that would be the pink point here. I'm showing this for beryllium dihydride as an example. And what we see is that its error is above chemical accuracy, but very importantly, its C0 depth, so its 2-qubit gate depth is something like 20,000 gates. So that's too large to be able to run on any reasonable quantum computer right now, or at least as far as I would have access to. But if we use adapt VQE, so these are two flavors of adapt, what you can see is that, first of all, the errors are reduced significantly, and the C0 counts are significantly reduced. So we're using the newest flavor of adapt. Really what varies between the adapt flavors is that they're using different operator pools. And so this is a specific operator pool that basically excites the same kinds of orbitals at the same time. And what you can see for CEO adapt is that it's bringing you down to, let's say, hundreds of C0 gates instead of 20,000 C0 gates for a very high accuracy. So that's the algorithm that we're using to build the ansatz. And what we've done is we've done this on, again, small systems, but I'll show you some of our larger results in a second. So here's water. We did water because we did the water dynamics before, and we ran this recently on IBM Fez. And this is just to give you a sense of kind of what we're seeing with this hardware. And again, we're not doing any post-processing on these results. I can talk about post-processing, but we're not. So we're using adapt VQE, and when we compute the circuits in a minimal basis for water, we end up with about 1,000 1-qubit gates and about 352 qubit gates that we need. And this is using, again, these coupled exchange operators. Without any noise or sampling, we get an energy of about negative 73.55 Hartrees. So that for those of you who know water, we're a little bit off because of minimal basis. And I mean, this is controllably off. We know we're off minimal basis, and also some tricks that we're using that I might have some time to talk about. Without noise and doing sampling, we can do about 3 million shots. And we get an energy around here. Now, when we run on FES, we can run with multiple trials. And the interesting thing to point out, which many of you may be familiar with if you've ever done chemical Hamiltonians, is that we're currently seeing fluctuations just because of the machine that are basically at the second decimal for Hartree, so at the centa-Hartree level. So for many people, you would recognize that is above chemical accuracy. So we're still not at the point where we're beating our classical machines by any means, but it does give us a sense of the kind of drift that we're seeing on these machines right now. And so we're working on different error mitigation strategies in order to bring this down. Let me also show you an example. I talked about this metaphosphate hydrolysis. We will also run that. I'll show you results with relatively shallow circuits. So this Hamiltonian, when we reduce it, ends up being about 44 qubits. And it roughly will take 182,000 polyoperators to execute. So we recently ran this on NERSC, literally in the past week. And it was really just to understand with a shallow circuit, what kinds of things come out. We're running with deeper circuits right now. So with a relatively shallow circuit and just sampling 50,000 shots, what we can take a look at is what states are we sampling. What we do is we typically start with the Hartree-Fock state, and then we expand our ansatz. So it's sort of not surprising that we get a lot of this state. The state over here is actually the Hartree-Fock state. But then as a result of running the circuit, again, a relatively shallow circuit, just to see what happens, we do sample some other states. So these are other sets of states that show up in our count. And then this column here is showing you everything else that shows up in our count. So we are able to get things that are off of Hartree-Fock, but of course not converged quite yet to chemical accuracy. So we're currently running these things with much, much deeper adapt circuits. So I don't want to take up all your time, but I'll just say a couple more things. Happy to talk about the details. So you might ask, how do we get to 44 qubits? And how is it that we'd actually approach all, full ATP hydrolysis? So we're using two key tricks to try to scale things up. And I think about it as puzzle pieces. What we can do is we can break our chemical systems up into fragments, but instead of them being independent fragments, we can actually embed them. And so we've been using tricks called bootstrap embedding in order to represent larger systems in terms of smaller fragments that we can actually model on a quantum computer. So bootstrap embedding was invented for classical electronic structure, I guess roughly a decade ago. There was earlier work on similar types of embedding before it. And the idea here is that you'll fragment your system into different fragments, let's say A and B. But what you do is you let these fragments overlap and you put these fragments in a bath. So the idea is you're going to solve the fragment Hamiltonian in the bath of other things. You're going to do this for overlapping fragments. And then what you do is you match those fragments. You match the density matrices, if you know what I'm talking about, of those fragments. And then you kind of iterate until you get a fully consistent picture of all the fragments overlapping and get the total energy. What this importantly lets us do is construct the full system's energy from little fragments. And so what we're literally doing right now is we're running that full ATP hydrolysis system using this quantum bootstrap embedding with little fragments and reconstructing the whole system. So this is like a long shot, moonshot goal, but this is what we're doing. We've already done it on the metaphosphate, and that's how we're getting smaller fragmented systems. I don't want to go on forever because I know you have a fixed amount of time. The other way we got to 44 qubits is using a variety of downfolding techniques. So if you're familiar with downfolding, we can basically contract virtual orbitals into a more active space and only pick a smaller subset of active orbitals that are kind of renormalized based on the virtual ones to produce smaller Hamiltonians that happen to be more dense, but yet they're smaller, and so you can run them on fewer qubits. So we've been putting these pieces together. And again, the whole goal and what we're after, and hopefully what we've shown, is that we can model these kinds of ATP hydrolysis systems. And so we're building up the quantum capacity, the quantum simulations to train these kinds of neural networks so that we can arrive at very, very truly quantum molecular dynamics networks that are highly accurate, but also thousands of times faster than QMMM. All right, so with that, I'll thank the team, and I do have to emphasize this is a team effort. I'm kind of an electronic structure theorist and biophysicist on the team, but people like Ryan LaRose and Norm Tubman, Sophia Kanamu have been helping with all the quantum computing, and others have been helping in other regards. So truly a team effort here, and I'm happy to take any questions that you might have. Thanks.